Good afternoon, everyone. Is my audio clear?
Yes, sir.
Can somebody type? Okay, thanks. So welcome to the continuation of transcription. We were discussing about eukaryotic transcription. I'll have a quick review of what we have learned in the last class on transcription, and then we'll proceed further. So the first things to remember, transcription happens within the nucleus and also along with replication also in nucleus in eukaryotes because they have a nuclear membrane. All of RNA processing and also happens in the nucleus. Ribosome formation happens in the nucleolus, right? There are three important things when we talk about RNA processing are the cap, which we will discuss today. The cap, removing of the introns and joining the exons, and also poly A tail. We'll see termination we will learn in the next class. Let's focus on today this. Eukaryotes have 3 RNA polymerases, whereas prokaryotes have only 1, of which RNA polymerase 2 is the most important or transcribed majority of the genes that result into that encode proteins. RNA polymerase 1 is mostly about the ribosomal RNA, and polymerase 3 is about transcribing tRNAs and small RNAs and so on, small rRNA basically. So there are several factors involved in eukaryotic transcriptional initiation. They are called as GTF or simply, general transcription factors. There are also other things involved. Those are called as mediator complex, and there are many other regulatory proteins which we will learn in the later classes, and also chromatin modifying enzymes. The core promoter, there are initiator, then you have downstream core promoter elements, DCE, that is downstream core element, DCE 1, 2, and 3. You also have Tata binding box, Tata box, which is almost similar to that of -10 sequence. You can see TAT, AA. It could be in this position, A or T, and so on. There could be some changes. So that is where Tata binding protein binds to Tata sequence, and there is BRE, that is TF2B binding or recognition element. BRE is for that, TF2B recognition element. So this is the initiator, that is where the transcription actually starts, and that would be the +1. And TF2D recognizes many sequences. In all these cases, there are also downstream promoter elements can also be there. Not necessary that all the promoter elements should be present in every promoter. Some may have, and some might be there, and some might not be there. So the general transcription factors, their job is they act something very much similar to that of prokaryotic sigma factor, that is to identify or recognize the DNA. So as such, the RNA polymerase, if you think of it as this, does not have a sequence specificity. Once it has to load onto the DNA. For it to load, it has to happen at specific sequences. If that specific sequence is what we refer to as the promoter. That promoter, if the RNA polymerase has to recognize and bind to DNA, the promoter should be recognized by some other proteins. those in prokaryotes it is something like sigma factor, whereas in eukaryotes it is general transcription factors. So they recognize the promoter and allow the loading of RNA polymerase. They're also involved in melting of DNA, that is formation of open complex. Right? What is open complex? When the DNA melts in some part, creating a transcription bubble, that is melting of DNA. Melting is basically denaturation in that part. So the DNA, so from closed complex, closed is when the melting did not occur. This is open complex. This would be closed complex. Then they're also, some of them are also involved in polymerase escape, or you can also talk it as promoter escape. The RNA polymerase that is bound here should move, and from initiation it should go into transcript elongation. So I hope you remember this picture. So we spoke about the first one, TF2D, and along with TBP, Tata binding box, they will bind here, and that causes a distortion. And then you will the loading of TF2A, TF2B also will happen. Then you have the loading of RNA polymerase, or TF2B will recruit RNA polymerase along with TF2F. Those two are kind of partners interacting. So you have something like this is the DNA, DNA binds to something, RNA polymerase binds to something. These two interact. That is how RNA polymerase is brought to the promoter. Okay. And when you have these general transcription factors along with RNA polymerase loaded onto the DNA, you call it as pre-initiation complex. Then we have the other factors, TF2E and TF2H, which has much more uh, straighter or profound activities. That is, it has helicase activity. Helicase is the one that separates the DNA, right? Like in DNA polymerase, we have read that breaks the hydrogen bonds. So with the helicase activity, it can form the open complex. Okay, and then there is also, we will discuss about this a little bit today, about polymerase has for promoter escape to happen, there is requirement of phosphorylation of the tail, and that is what will allow the RNA polymerase to detach from the general transcription factors and move on to the elongation phase. That is promoter escape. So promoter escape, as I was describing, it is caused by the phosphorylation of the carboxylic terminal domain of RNA polymerase. So it has several repeats like this. The tail has a sequence. We will get to this in another slide, but it has the sequence. In each one, you can recognize, you can think of YSPT, SPS. That sequence is there. That can be phosphorylated by the kinase. One subunit of TF2H, it has helicase activity, but it also has kinase activity, because of which it can phosphorylate the RNA polymerase CTD, and that will allow the promoter to escape. So to remember, you can think of it as this TBP is there are numerous general transcription factors. We didn't have to discuss all of them, but you can remember that there are several subunits. Each one could be a complex of proteins. So TBP is associated with about 10 TAFs. They have some homology to histones. When you say homology to histones, the sequences seem to match. Some similarities at the level of sequence, and sometimes because of sequence, it could also be because of structural similarities. Okay, and when you're doing this, I would want you to remember TF2H, TBP, and TF2B and D also are important. Just you can learn from this. And I was describing this is a good way of how you try to make your notes in case you want to remember just before an examination. I'll skip this. And in reality, when transcription is happening within the cell, there are, in eukaryotes especially, we have learned that they are all in nucleosomes like this. And DNA, if transcription has to happen, there should be some remodeling required. For that, you will have several complex interactions other than the general transcription factors. These are the general transcription factors, but there are also a complex of proteins called as mediator complex. You also need chromatin remodeler, you need um, modifiers, and so on. And proteins like activators, which may increase the rate of transcription initiation, but there are also repressors, which may prevent transcription. The mediator complex itself is, as shown here, it's a huge protein complex made of several proteins. And if you see in comparison of yeast mediator with that of human mediator complex, you will see that human mediator complex has more subunits. In elongation, so the next phase after promoter escape is what we describe is as the elongation. Elongation again has its own set of elongation factors. There are two important activities of elongation factors. One is to stimulate elongation, and the second one is proofreading. We discussed this in the last class. So they are recruited at the CTD, carboxyl terminal domain of RNA polymerase, and they are they will keep increasing. Firstly, I would think you have to think of TF2S, that is shown here. Here is a comparison between two different elongation factors. In E. coli, we learned briefly about GREB, and the function of which is involved in elongation, stimulation of elongation, and also it is involved in along with NUS protein, it's involved in proofreading. Okay, in eukaryotes, the similar function is taken up by what we call as TF2S. Okay, they can fit into the channel almost very much similar. Yes, you have to realize that the RNA polymerase of prokaryotes and eukaryotes, they may have similar structures in the active site, but there is a large amount of difference as well. Let's not go into the structural details, but I hope you understood the point. So the second is to say basically, if RNA polymerase is moving along the DNA and it is transcribing, TF2S is taught to, it is like something that is telling move, and it is making RNA polymerase to move further by not allowing it to pause at that location for too long. So the pause time is probably reduced, and that responsible, it is TF2S that apparently reduces the pausing time, which means you're actually increasing the rate of transcription. There are also, I think in the last class we also learned about, I will get back, but let's see. There is one more thing during elongation, is that, as we are saying, as we were discussing, the DNA is present as nucleosomes, right? It is wound around histones, and if RNA polymerase has to move, the DNA should be unwound from the nucleosomes, and then DNA that is double stranded should be made into this transcriptional bubble like this. So there are two activities. In prokaryotes, we don't have that much of a problem. All we need, the DNA is, if not all, most of it is free, it is completely like this. All the RNA polymerase has to do is just keep denaturing it transiently and then making a RNA copy. In eukaryotes, you have to dissociate the DNA from histones, and then also using helicase activity should unwind the DNA and then polymerize. So there is something called as FACT, that is a factor which is apparently these two together, SSRP1 and SPT16. They are involved in displacing the histones from in this direction. Basically, you can see here, you can think of it as the H2A H2B dimer is bound to one of the SPT16, and after the RNA polymerase has moved on, the the fact or the SSRP1 SPT16 would put back the subunits back into the nucleosome, right? These kinds of, this is still not entirely understood, but it seemed to be that these, the at least partly it is indicating that H2A H2B dimers, or maybe for other histones also, you may have different complexes that bind and not the the point or importance of this is, if RNA polymerase was moving, the histone is released outside, and then again, where should the histone form and how does it come in here, right? Instead, if there was a factor, that factor would allow you to only the factor would allow you to keep the histone within the location, then it is easy to reorganize the nucleosome. That is the essence of this slide. So I will try to describe something about eukaryotic RNAs. Eukaryotic RNAs, there are three things essential. You know that the genome, sorry, the DNA is something like this, okay, and it has exons, introns, and exon, intron. I'll just draw two introns, and this is an exon. 1, 2, 3. And if you compare it, if upon transcription, you will get RNA. RNA has the exons. I'll just draw only the exons, 1, 2, and 3. There should be processing that should have happened, and that is, processing is the joining of the exons, thereby you are eliminating the introns, 1, 2, and 3. In addition to that, the RNA that is being produced should be stable when it is sent into the cytoplasm, and it should be recognized by the translational machinery. For that, you need a cap that is called a G cap. This is the 5' end of the mRNA. And you need to have a poly A tail, which we will describe later, at the 3' end. Right? So there are three important things that happen to eukaryotic mRNA. You have capping, there is splicing of exons, and polyadenylation of the 3' at the 3' end. And say if there is a DNA and RNA polymerase has just started, promoter escape has happened, and you have the RNA coming out. As the RNA polymerase is moving forward, you will have the production of more and more RNA. So the first thing that comes out is the 5' end. And what is the, if we look from this direction to this direction, okay, we will look at this place where here, if you see that, we can identify what should be done in each of these locations. What is the first event that should have happened? If you compare it with the this is the mature RNA, and this is the nascently or premature one, you can think of it. Assume that transcription has started here and RNA polymerase is moving in this direction. What is the first thing that would come? The 5' end will come. What should happen? What is the modification that is required in the 5' modification? You need a G cap. Then you have the formation of more, and then you have exon 1. And then here you are at the end of an exon 1. And then there is an intron. This intron should be removed. So what is the next set of proteins that you require as transcription is progressing? You need to recruit proteins that are involved in removing this intron. What's after that? Exon, nothing to remove. And after that, again, you have another intron to be removed. So you have to recruit another protein, another set of factors involved in removal of the intron. And at the end, you will have a poly A tail. We will discuss this signal for a polyadenylation. That is where termination will happen. When we read about termination, we will discuss this in detail. But once the RNA polymerase crosses the signal and termination has happened, you have to recruit a protein that will add multiple poly A tails. What I'm intending to say here is there is a sequence of events as the transcription progresses, and in each case different set of proteins are involved. GCAP proteins, intron removal factors, and polyadenylation. They should happen. You can also think of it like this. What would happen if first polyadenylation protein is bound? First of all, how do you ensure that they bind there? That is where you want to learn about the CTD, carboxylic terminal domain of RNA polymerase. It has a long tail and it has several repeats of YSPTSPS. Y is for tyrosine, serine, proline, tyrosine. Sorry, this is threonine. This is tyrosine, TYR. Y for tyrosine, T for threonine. S is serine, P is proline, and S is serine. So there's a single letter coding of the amino acid residue present in that C-terminal domain. And you have several things. The meanings of there are all these amino acids are one second. All these amino acids have tyrosine has a OH group. You have serine is also having an OH group. Proline is different amino acid. Threonine has a hydroxyl amino acid. So is serine and serine. All these positions are likely or good substrates for phosphorylation by specific proteins. So if this particular sequence is not phosphorylated, there is a meaning to it. If only the serine in the fifth position is in the fifth position it is phosphorylated, there is a different meaning to it. If you have in the serine 2 is phosphorylated in the second position, then there is a different meaning. Okay, so the first thing is you should understand is here assume that you have RNA polymerase, it is first starting to synthesize. If you have two phosphorylations like this, okay, let me talk to you about this before. Fifth position, serine is phosphorylated. It has two meanings, two things. At what is happening in the DNA level and what is happening to the RNA that is coming out. Okay, it means if there is no phosphorylation at all, that means pre-initiation is just happening, formation of pre-initiation complex. Then TF2H with its kinase activity would phosphorylate this serine 5, and because of that phosphorylation, now RNA polymerase gets a signal that it is now moving or escaping the promoter. That means it dissociates from the general transcription factors and will start transcribing, moving on from initiation to elongation. At the same time, this is also a signal for capping. Capping at the level of RNA processing. This is at the DNA. Let me write it again. So you can, what here it is, I hope you remember what it means by histone tail modifications, right? Histones can be acetylated, methylated, and also phosphorylated, and each of them have their own signature. What it means to be acetylated, which is often associated with transcriptional activation, methylation, sometimes repression, sometimes activation. It depends on the context. In a similar way, you will also see that the changes or the post-translational modifications of RNA polymerase CTD tail has a meaning. Phosphorylation, no phosphorylation, pre-initiation complex. RNA has not come out. That's why they put a bar here. In the second position, second one depicted here, serine 5 is phosphorylated, which would mean that you have escape, that is, you're escaping the promoter. And the first event that should have happened in this one, as I was describing, is the capping should have happened. So it is also signal that capping should happen. So what happens is the capping enzyme binds to the CTD tail. So when the RNA emerges completely out of the RNA polymerase, the capping enzyme will jump onto the RNA polymerase as shown here, and then it will do the capping. G prime. We will discuss about the cap in a while. And there is now change in the phosphorylation state. Once this promoter escape and capping enzyme have done, then you have the serine on the phosphorylation on the S5 is removed, and serine in the second position is phosphorylated. That has a different meaning. That means now you are ready to go or proceed to elongation. And you also would need a lot of proteins or factors that are important for splicing. So once the phosphorylation status of the tail has changed, capping enzyme has moved on, you have a different thing. And now you're going to get the splicing machinery that is coming here. As soon as an intron, the exon, end of an exon comes, this protein jumps to that place. And that keeps on happening until you are reaching a termination. When you are getting to a termination site, you will have signal for polyadenylation. And when that polyadenylation signal comes, the proteins, the enzymes required for cleavage and polyadenylation will jump onto the RNA. And that is how the RNA, while it is being produced in eukaryotes, will keep on changing. First, capping. Second, removal of introns and joining of exons. And third is polyadenylation. In prokaryotes, you should remember comparison between them. I have my favorite question is what are the differences between prokaryotic and eukaryotic mRNAs? You can try to find it out if you can. Remind me if we don't discuss this in the future. So what is the cap? About splicing, we won't discuss much because we have a whole chapter to discuss in the next class. But in this one, we will discuss a little bit about cap. That is also my favorite topic because I want you to learn some structures. And it's also interesting. Why do you need a cap and what is the importance of it? So this is the 5' end of the RNA that is freshly synthesized. And you also, I hope, remember that usually with the +1, it adds a, and then you'll have other bases. So here it is shown as A, and on the 5' side, it has 3 phosphates. You'll have alpha, beta, and gamma phosphates, right? And then you have the remaining bases in this direction like this. So there is an enzyme called as RNA triphosphatase. That means the RNA, this is a one of the RNA molecules, if it has, assume this is the RNA and this is the 5 prime and this is the 3 prime. The only place the RNA would have 3 phosphate groups is on the 5 prime end, because that is the first base added, first nucleotide. RNTP has come, it has 3 phosphates, it is just retained like that. Now look at the enzyme, it says RNA triphosphatase. So this is the these are the 3, or this is the triphosphate, and phosphatase means the one that degrades it. It will break the first, the gamma, right? This is alpha, beta, and gamma. It removes the gamma phosphate. Okay, the enzyme is RNA triphosphatase. And then the beta phosphate joins with the alpha phosphate of a GTP. This is GTP, and GTP, the beta and gamma phosphates are eliminated. Excuse me. Sorry. Yes, sorry. Beta and gamma are eliminated. So the beta of the RNA, this is the RNA, this is GTP. The beta phosphate of RNA joins with alpha phosphate of GTP. And now you have 3 phosphates again, of which this is alpha, this is beta. They belonged to the RNA. Actually, they belong to the RNA that is being produced. Whereas this alpha belongs to the GTP here. But this is a 3' phosphate. And this is called as 5' linkage. Why is that 5'? This is because you have G. If you look at, this is the 3', 4, and you have 5, right? 2, 1. 1, 2, 3, 4, and you'll have a, sorry, wait a second. I have, yes. So if I draw it this way, it's easy for me, this one. So 1 prime I should call, 2, 3, 4, and this is the 5th one. And phosphate is here. Assume that this is the GTP one. Okay, I'll write 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And this is phosphate alpha. And of that, the one that of RNA could be something like this: 1, 2, 3, 4, and this is 5 prime, 4 prime, 3 prime, 2 prime, 1 prime. And I will write A here because to indicate that this one. So the 5 prime carbon would have had 2 phosphates, because one of the phosphate, the gamma phosphate, is removed by RNA triphosphatase. And so this is alpha and this is beta phosphate. This is joined, the beta phosphate is joined to the alpha phosphate. So what I'm trying to say, to mean what it means to indicate that it is 5 prime. This is bound on the 5 prime, and this phosphate is also on the 5 prime. That's why you call it as 5 prime linkage. When we go into splicing lesson, we will find a few more, or a couple of more linkages that we will discuss at that time. Okay. So first, RNA phosphatase, removal of gamma phosphate from the RNA. Then guanylyl transferase, the enzyme that mediates the interaction between beta phosphate of RNA with that of alpha phosphate of GTP, is called as guanylyl transferase. Then there is also furthermore modification that is done by methyl transferase. It is simple to say. There is a methyl group added, and it's at the 7th position of guanine. Okay, so The capping, 5' capping happens in 3 steps. Removal of gamma phosphate from RNA, mediated by RNA phosphatase. Binding of beta phosphate of RNA with that of alpha phosphate of GTP, and that is mediated by guanylyl transferase. And the third one is methylation of the guanine, GCAP, that is on the 7th position, and that is called as that is done by methyl transferase. Okay. This is a structure that I would want you to remember, and I could ask you for this structure with numbering. So it is one that will allow you to understand things much better, and also memorize a little bit of the of GCAP, because of which you will also remember some of the chemistry or the bonds or structures that are involved with nucleic acids. This is alpha beta of RNA, and this phosphate would be the alpha of the GCAP. Here is the methyl group. This is the same thing, except that you can remember some of the enzymes. RNA, this is from a different textbook, so you might have a little bit of difference in terminology. It's all right. So polyadenylation, I will see if there is, yes. When the RNA polymerase keeps on moving, and there is a certain sequence that is called as poly A signal sequence, and that is present within DNA. When RNA polymerase goes past this, that means it has transcribed the poly A signal in RNA. There are two proteins called as CPSF and CSTF. So they have certain function. Each of them, one of it is the one that cleaves the RNA somewhere like this, and there is a product called as a protein called as CSTF, which stimulates the cleavage activity of CPSF. Okay, CPSF, the names are C cleavage and polyadenylation specificity factor, and CST is cleavage stimulation factor. So CPSF has two functions. One is to recognize the sequence, poly A signal sequence in RNA, and the second one is to cleave the RNA downstream of it, somewhere like this. And CTCF stimulates this activity. So upon RNA cleavage, CTCF is removed, and then there is a protein recruited to that site. It is called as poly A polymerase, or PAP, as you understand from the name, poly A polymerase. All it does is add more adenines. That's the only work that it does. And the poly A, it binds to the end of the where the CSPF has made the cleavage. It binds there, and then the PAP, poly A polymerase, adds more and more adenines, and that is the poly A tail, which could be in hundreds. Hundreds of adenines. Okay. And because you have a poly A now trail, there are certain proteins called as poly A binding proteins. They bind to the 3' end. By doing so, there are two important things that happen. First thing is that the 5' the GCAP, and you have poly A tail. The GCAP and poly A tail will, the important thing is that they will provide protection to the 5' and 3' ends. The cytoplasm in general has a lot of RNases. Working with RNAs is very difficult, because they're very fragile. You change the temperature, you change a little bit of pH, they will die, they will get degraded because of the 2' OH. And there are also RNases from our body and many things. They might come and contaminate your RNA samples, and that is where RNA could get degraded very fast. So working with RNA is actually one of the technically very demanding, in the sense that you have to be very, very careful so that it doesn't get degraded. Why I'm saying that is that if, because majority of these RNases could be exonucleases. What do you mean by exonucleases? They will degrade from the ends. So if we want the RNA to be stable, sorry, we want that the 5 prime end and the 3 prime ends are safe. And here are some of the evolution, what evolution has come up with: G cap and a poly A tail. And they are also the same. These modifications are also recognized by a translational machinery, and that is another role that these proteins play, or these modifications are important for. With the termination, so what happens was we have a protein, the cap, 5 prime cap is added, the poly A signal is made or synthesized, and the poly A tail, because of the cleavage, will be done, and PAP will recognize and will synthesize poly A tail. Right? Whatever is happening with this RNA, it's over. What would happen to this one is the question. The RNA polymerase is still transcribing as if nothing has happened. So that is where there is a protein complex, or which is called as RAT1 HXRN2, which binds to the naked uncapped end. Any RNA polymerase, it is design itself is like this. If a RNA is coming out of RNA polymerase and it doesn't have a cap, that means it would mean that the cap, the that is a part of the leftover part of the RNA. The other part with the cap and poly A tail is left off for maturation or further processing. So that is kind of signal for this RAT1, which will bind to the uncapped end of RNA and will degrade a lot, and eventually the RNA polymerase will dissociate from the DNA. So there could be some more stimulation because of loss of RNA and so on. And some people also think, or this is in an allosteric model. There are two models. One is called the torpedo model, and the other one is called as allosteric model. While there could be differences in different organisms or transcriptional termination of different genes, it is just thought that if the breakage of the RNA happens, that itself induces some changes within the conformational changes within the RNA polymerase, because of which it will dissociate. Okay, you call the other one as torpedo model is because you have this factor which is attacking here and eventually destroying the association of RNA polymerase with the DNA. So the last two slides, yes, I will finish this and then I'll stop. So we didn't speak about RNA polymerase 1 and also about RNA polymerase 3, about the promoters. So in the promoters that to which RNA polymerase 1 binds have two important components. One is the core promoter, which is very much similar to that of what we have seen in promoters of RNA polymerase 2. And there is also another element about upstream minus 100 to 150, minus 150. That is called as upstream control element, UCE, to which there is a complex like SL1 and UBF, which is along with TBF, TBP, that is Tata binding protein, binds and initiates transcription using that is done by Pol1. Okay, so Pol1 recognizes. That means this promoter is present upstream of several of the rRNA genes, rRNA genes. So you can assume that this is the gene for rRNA, and I'm not putting an arrowhead like this. What does this arrowhead, when you say like this, it would mean that it is an ORF. Typically, that's what it means. The rRNAs genes do not produce a protein. They only produce a RNA molecule. Here they are producing rRNA. That is the final product. Sorry. So that's about the promoter element of for Pol1. Similarly, for Pol3, there is a little bit difference, and it could be downstream. When you say it is downstream, what you mean? You're counting from +1. The broken arrow shows it is the first base that is being added. And where are its promoter elements? They are after the start. It's like something binds here and then stimulates, or many others are also involved, but transcription starts from this place. Okay, that's why these promoter elements, which are referred to as box A and box B, they're downstream of the initiator region, but they also allow the binding of TF3B. We are calling as TF3B. I think I forgot to mention this. Why do we say TF3B, 3C, and so on? Because they are transcriptional factors for polymerase 3. In the previous one, we mostly spoke of TF2B, TF2D, TF2H. We're talking about transcriptional factor for RNA polymerase 2. Okay, here we are saying TF3B because it is for the RNA polymerase 3. So there is a complex TBP. The complex protein is TF3C that recognizes box A and box B. You have Tata binding protein, and you have 2F3B. And they bind, and polymerization or transcriptional initiation starts from here and proceeds towards the end. Okay, I think that is the end of the class. Typical questions which are usually straightforward. Right? These do not have any twist or thinking. It's mostly about recollection. If you remember the classes that we have discussed in the first classes or so, I told you the levels of learning. I put this is the bottom level is remembering, then understanding, then applying, and to make it, there are other levels, but I will just say it as creating. We will just consider 4 levels. These questions that I've put here is remembering. What is the mechanism of prokaryotic transcription and proofreading? It's like you have to you should have read it and recollected it and reproduce the answer. What is the mechanism of eukaryotic transcription? Everything is the same. These questions will only be at this level, remembering and at the most understanding. But when you get your questions, because it's also an online question, you will have application involved in it. We will practice some questions for every lesson, and I hope you will be able to do it to your best possibility. And this is also, I told you one of my favorite question. Where is it? It's not here, I think. Okay, there are differences. That is one thing you have to know. Types of prokaryotic promoters. What I said is the differences between the RNA of prokaryotes and mRNA. You can think of mRNA of prokaryotes and eukaryotes. I will just use 1 minute to give you some idea. In prokaryotes, you call them as polycistronic. And in eukaryotes, you call them as monocistronic. I'm talking about the DNA. So this is a good time to introduce, so I will do that now. In prokaryotes, there is a promoter and there are genes, gene 1, gene, okay, there will be a gap here, okay, that is intergenic region, gene 2, gene 3, gene 4. They all will be transcribed as one mRNA. So gene 1 is here, gene 2 is here, gene 3, and gene 4. That means they are all open reading frames. This will produce one protein, and so upon transcription, ribosomes will take up translation. Ribosome will be loaded, and it will start reading from here. So each of these open reading frames will produce one protein. Sorry, each one will produce. This is one mRNA with multiple open reading frames, or if you talk in this sense, this is one piece of DNA which is having as one transcriptional unit, right? From here, transcription starts here. This is +1, and here is the terminator. So it is producing one mRNA, each having multiple open reading frames, and each open reading frame is producing multiple, one protein each, right? That is why you call it as polycistronic. Cistronic is almost similar to that of a gene. Cistron is almost equal to gene. Poly means many, many genes. And a transcriptional unit arranged like this is called as operon. Right? In prokaryotes, you will find that many genes are expressed as operons like this. In eukaryotes, however, they are monocistronic. That means they should have one promoter, there will be one open reading frame or a gene, and there will be a terminator. So this will produce, upon transcription, it will produce one mRNA. It will have one open reading frame, which will produce one protein. But it's not straightforward like this, right? Each of these genes have introns, right? With exons, exon 1, exon 2, exon 3. And it would be produced as a pre-mRNA with the exons, with the, introns. And upon splicing, you will get forming the mature mRNA. I hope you see the differences between prokaryotic and eukaryotic mRNAs. So what else is different? I'm not going to take too much time. Okay, time is up. Yes. So there will be a cap, G cap. There will be a polyadenylation tail. There is splicing that has happened in eukaryotes. In prokaryotes, no G cap, no poly A tail. In prokaryotes, they are polycistronic. In eukaryotes, it is monocistronic. Yes? I'll stop here. We'll meet on Monday again. All the best. Have a good weekend. Take care. Bye-bye.